Hi everybody, this is Mrs. Lucci. Sorry I'm out today, but hopefully you will have a very productive class period today. So in terms of today's focus, we are going to be taking a look at the cell cycle today. Last night, the homework was block 3, and you had to complete pages 7 and 8 in the packet. So we'll be going over the homework on pages 7 and 8 today. And then from there, I have two follow-up activities. One is a link that I've put on Google Classroom for you, which gives you a nice true view of how the cell actually appears in all of those stages of the cell cycle where the cell is trying to divide from one cell to two. The second thing that I have is the handout that you picked up on your way in that's titled Chapter 2.3 Reinforcement Radio K Cell. It's kind of a fun way to look at the descriptions of the cell cycle and mitosis. So it's a good activity to have you do in class. The homework for the weekend is block 4, which is that last section on cancer. When you come in on Monday, we'll be working with the microscopes to actually view the different stages of the cell cycle. So doing the activity today on the Chromebook through the link that I've put on Google Classroom will be helpful for you, because then you'll see how cells might look in different stages of the cell cycle, and it shows not only plant but also animal cells, so you get a good sense of what it looks like in the real world rather than just a drawing that you have in your textbook. So I really like that. Remember to use your Chromebook numbers. When you're taking the Chromebooks, students should not be putting them in or out of the cart, and students should not be plugging them in and out. So just return them so that the substitute can slide them back into the cart for you at the end of the class period. All right, so homework. You should have your homework packets out in front of you. As I said, last night was pages 7 and 8. So I'm imagining you might have some questions on number 1, which is actually the cycle, or the cell cycle, which is all of those stages. So in fact, if we're taking a look at the cell cycle, right, the very first stage is interphase. And in interphase, as it says, the cell grows and makes a copy of its DNA. From there, we go to the stage called Mitosis Prophase. And in Mitosis Prophase, what happens is we see the fine strands of DNA starting to weave themselves up into chromosome. DNA is long, thin strands, so it really would be very difficult to take all these long, thin strands and try to evenly divide themselves up in two brand new daughter cells at the end of the cell cycle. By having the DNA essentially knit itself into a chromosome, it makes it a lot easier to split it into two new cells and make sure that each new daughter cell gets an exact copy. Okay, so that's actually mitosis prophase. Okay, remember that first stage was interphase, and I know that one's filled in for you. Interphase is generally the stage that most cells are in as they all grow to maturity, and that can take a long time. Once the cell is mature and it's received a signal to make a copy of itself, then the DNA will start to be copied, as well as all of the organelles, because you can't have one cell get all the mitochondria and another cell get all the Golgi bodies. So all of the organelles will have to be copied in addition to these these organelles called centrioles. Okay, so that's interphase. Then we've got prophase. Okay, from there we go to metaphase. Okay, metaphase I always remember because it starts with an M, and so does the word middle, and the chromosomes line up along the middle of the cell in metaphase, and they're attached to these spindle fibers. I often think of the spindle fibers as kind of like train tracks. They're a guide for which way the chromosomes are going to go. Now you'll notice here that these chromosomes lined up along the middle of the cell are actually paired up. So if you look at them here, we've got red and blue chromosomes, but it turns out this blue chromosome has 4 legs, the red has 4 legs, right? And these all have 4 legs because these are already copied. So you can split them apart, which is what's happening in anaphase. And this half of the chromosome is identical to this half of the chromosome. Because they were copied way back in interphase and then wound together into a chromosome, now splitting them apart makes sure that this soon-to-be daughter cell has the same genetic information as this soon-to-be daughter cell. So this is happening in anaphase, okay? That's the description that we have down here, anaphase, okay? Next, we have telophase. So in telophase, we've now split the nucleus, or split the chromosomes, I should say. So now that there's identical chromosomes in each half of what will soon be 2 cells, but right now is still only 1 cell, we now have 2 nuclear membranes forming around the chromosomes. This is going to help create 2 nuclei. So by the end of telophase, you still have 1 cell, but it'll actually have 2 nuclei. Okay? Last but not least, we get to the last stage, which is, in fact, cytokinesis. Okay, so cytokinesis, finally, we're getting this total pinching in of the cell membrane, and it splits apart, and we have 2 brand new daughter cells. We started with 1 parent cell, it splits into 2 and becomes 2 new daughter cells, each of them with about half the organelles, but identical sets of chromosomes and genetic material. One other thing you might notice is that in a lot of these descriptions, right, not interphase, but in metaphase, in anaphase, telophase, right, and wait, up here, oh, no, not that one, but in prophase, the one that we saw before, all right, in prophase, they all started with the term mitosis. They started with mitosis, so it was mitosis prophase, mitosis anaphase, metaphase, telophase. And the reason for that is mitosis is often described as the second stage of cell division, interphase being the first, mitosis being the second, and then cytokinesis being the last. Mitosis is actually 4 steps, all devoted to dividing the nucleus. And that's because the nucleus is so complex, it takes 4 steps just to do it right, because that DNA is so crucial to the survival of the cell. Okay? Hopefully that's a good refresher for you for that first question. Okay? Now, as it turns out, if you think about what takes the longest, that's actually going to be interphase. And the reason for that is the cell has to be completely mature before it can start dividing. So it can take a while for a cell to reach maturity, right? In terms of when we're copying the DNA, that's also during interphase, as it says in the description here. So we've just gone over the table and the first 2 questions, okay? Now we are on question 4, okay? So if we look at question 4, during which stage does the cell membrane pinch the cell wall into 2? During that stage, we're actually talking about pinching into 2 cells. That is going to be cytokinesis. The difference between plant and animal cells is that obviously animal cells don't have a cell wall, so they are not going to need to build up these furrows to create a cell wall, right? So we're thinking of cytokinesis here, right, as the stage when it finally pinches off into 2. And then animal cells, as we said, do not have a cell wall, okay? All right, next up we've got question 5. This is an interesting one. How do DNA molecules pair up? Why is the strand identical? So I've got a good visual for this, right? Your book shows you an image of DNA that looks something like this, although probably a lot smaller. If you look at this image of DNA, what you'll notice is that everything seems to be color-coded. And in fact, if we take a peek here, I'll just kind of drag this down, you'll notice the thymine is the red one, adenine is green, guanine purple, cytosine yellow. And that's really just for the purposes of providing it a color to make it easier to identify. But if you notice, more importantly, the shapes of these different nucleic acids, right, these bases, you'll notice that the adenine only bonds with the thymine. Okay, so this is that adenine that can only bond to the thymine. If you tried to stick this adenine onto the cytosine, it's not going to fit, right? So we usually just abbreviate it A and T, C and G. Okay, and that basically means that because of the way the nucleic acid bases are structured, adenine will only bond with thymine and cytosine will only bond with guanine. So how do the nitrogen bases pair up? Well, adenine pairs up with thymine and guanine pairs up with cytosine. Okay? Since they only pair up one way, the DNA makes an exact copy. Okay? So that brings us to question 6. If this is your sequence, what's the other half of this double strand going to have to be, right? So if it's AGA-TTC, it would be TCT-AAG, okay? And that's the only way it can match up, okay? Number 7, really this is one that you're just creating for yourself with the letters I, P, M, A, T, C. And I've had a lot of different answers. One that I've liked in the past is I proudly make a terrible cake, right? And that's a great one because it uses all the letters and it puts them in the correct order, okay? But those are basically all the questions that we did for homework, okay? So one last thing just to kind of reiterate this whole idea of why DNA copies are so identical to each other has to do with the idea, not this page, but it has to do with the idea that the DNA, when it goes to make a copy of itself, will actually begin to split apart, right? And as it splits apart, one half of the strand will pair up with these loose DNA pieces, and with these loose DNA nitrogen bases, and those will be used to attach to each half of the strand and create identical copies of 2 new DNA strands for the new cell that is needed so that each cell has an identical nucleus. Okay? Hopefully this is helpful for the homework review for you. At this point, now that the homework has been reviewed, you can get onto Google Classroom, go to the link, all right, and what you'll find once you go to Classroom, I put the link right at the top here, so it's the next announcement, and you'll see that we have stages of the cell cycle as a model here, and you just have to decide what this closely matches and then put it in the right place. And then they'll give you new stages every time, and there'll be lots of examples, some from animal and some from plant cells. Hopefully this helps. Bye-bye.